Now, let us see the various steps involved in the protein synthesis. We had seen the two key players, that were the tRNA and the ribosomes. Now, the first step involves the charging of that very tRNA. What it is, we will come to that in this lesson, which is dedicated to the step 1. But before that, let us see what is present in step 2, 3 and 4. Step 2 involves initiation. Now, this step is similar to that we saw in transcription. Step 3 involves elongation, yet again similar, but the process altogether will not be similar. And last one is termination. And because all of this is going to be carried out by reading specific sequences, somewhat mechanism would be similar, that there would be a sequence that would identify an initiation of protein synthesis would begin, elongation and termination. Over here what we are dealing with is amino acids, alright. So, we are going to see these 3 steps in subsequent lessons. What we are seeing here, let's talk about that. Now, what do we talk about that? Charging of tRNA involves the addition of amino acid according to the anticodon loop sequence at the 3' end A base pair of tRNA. The 3' end of A joins the amino acid, okay. The A that is present, A is what? A is the base pair that is present, or the nucleotide that is present at the 3' end joins the amino acid, the specific amino acid, and that is known as charging of DNA, or otherwise known as amino acylation. Amino is alright, it is taken from amino acid word. What is this acylation about? Now, this charging of tRNA and what this acylation is, you will get to know when you understand the process in 2 steps. The first step involves the binding of amino acid in the presence of, with ATP, sorry, not in the presence of ATP. Binding of amino acid with ATP in the presence of a highly specific enzyme known as amino acyl tRNA synthetase. This is a highly specific enzyme for each amino acid. So, it identifies which amino acyl tRNA synthetase would combine with which amino acid. In the presence of ATP, it would give rise to amino acyl adenylic acid. This is simple chemistry that is at your disposal, and 2 phosphates would be there. So, the ATP's adenine combines with the amino acid in the presence of this particular enzyme, that is amino acyl adenylic acid is being formed. Now, this amino acyl adenylic acid would combine with the specific tRNA. Again, the specific tRNA, it would combine, it would give rise to charged tRNA that has the amino acid attached to the tRNA that we have over here. So, in this way, charging of tRNA takes place. Of course, the phosphorus would combine with this adenine, and there would be amino acyl tRNA synthetase that would come out of this, that would be regenerated. So, what we have achieved in these 2 steps is that the amino acid that was present in the beginning, that is combined with tRNA. tRNA has enabled the joining of amino acid at its 3' end, okay. So that is what charging of tRNA is, and this is the very first step of translation that you have to remember. I hope it is clear. Now the next step would involve the step of initiation.